group of plants, I study two native pine species in California, so I'm a bit biased towards gymnosperms. But let's get started. So just a quick review of basal land plants. So the basal land plants include the non-vascular land plants. Those were the liverworts and the mosses, as well as the seedless vascular land plants. So what we looked at last week. So with these guys, they utilized their spores for dispersal. So with spores, they were basically dormant until water allowed them to germinate and produce a brand new gametophyte. So that spore had to be in some kind of really moist and wet environment in order for that life cycle to complete and essentially finish. So once the spores landed in water, those haploid spores would then produce the brand new gametophyte, the brand new plant. So in this case, gametes would be produced. So that would be the archegonia and the antheridia. Remember that the archegonia is female and the antheridia is male. So the archegonia would house the egg and the antheridia would house the sperm. Those two gametes would come together. They would undergo fertilization to produce a diploid embryo. And then that diploid embryo undergoes meiosis, produces a diploid sporophyte, and then that diploid sporophyte will produce the haploid spores. In the case of mosses for non-vascular land plants, remember that the spores were housed in the capsule or the sporangium. With seedless vascular plants, that could either be on the sori, for example, on leptosporangiate ferns, or it could be on the strobelites, so the cones. So it all depends on what the species is. But remember that spores essentially required water or moist habitat in order to produce that brand new free-living gametophyte. So they had to land in water in order for that life cycle to essentially finish. So this is what it most likely looked like millions and millions of years ago. So large body of water, we have what looks like some moss over here. And again, they really needed that wet and moist environment in order for those spores to be dispersed. Here's another picture of what it possibly looked like during the Ordovician. So these, as you can tell, these plants are a lot smaller compared to what we see now. Seedless vascular plants and non-vascular plants in general are a lot smaller, especially because they don't have secondary xylem. Now, although seedless vascular plants do have true xylem and phloem, they don't have secondary xylem, meaning that they don't have wood or bark, so they really couldn't get that big. There were species in the Lycopodiophyta that could get really, really tall. One of them was Lepidodendron, but that is extinct now, so you don't need to worry about that. But those got really, really tall even without secondary xylem. But for the most part, seedless vascular plants and non-vascular plants were really small. So if we look at this phylogenetic tree right here, over here on the left, we have all of our basal land plants. That's what my cursor is on right now. And then over here on the right are the more derived groups. So right here, the naked seed plants or the gymnosperms is what we're going to be focusing on today. And then all the way on the right is what we're going to be focusing on next week, which is the angiosperms or the flowering plants. So this is what you guys are most likely more familiar with. But here on the left, again, we have our basal land plants. Right here is Hepatophyta. That's the old name for the liverworts. We refer to the liverworts now as Marchantiophyta, but same thing, Hepatophyta, Marchantiophyta, both referring to the liverworts. Then we have the Bryophyta, so those are the mosses, and then Lycopodiophyta, so that would include the lycopods and Selaginella. And then the last one is Pteridophyta, or Pteridophyta. I don't really know if I'm pronouncing that correct. But this is essentially the Manilophytes. That's just the old name. So if you see Manilophyta or Pteridophyta, it's referring to the same group of species, but this would include your leptosporangiate ferns, aquatic ferns, horsetails, etc. So these are all your basal land plants. In between the evolution of the basal land plants and the seed plants, you have 4 characteristics that evolved that allowed gymnosperms and angiosperms to really outcompete and succeed over these basal land plants. And those 4 characteristics are seeds and pollen. Seeds and pollen evolved at around the same time. They kind of go hand in hand with each other. And then you also have the evolution of secondary xylem and phloem. Now remember that seedless vascular plants, so Lycopodiophyta and Manilophyta, they do have true vascular tissue, xylem and phloem, but they are not secondary xylem or secondary phloem. So that's why gymnosperms and angiosperms are typically a lot larger compared to these basal land plants. And now we also have the evolution of the gametophyte being housed on the sporophyte. So now the sporophyte is the longer lived or the dominant generation, and the gametophyte completely relies on the sporophyte. It's essentially not free living anymore. It completely relies on the sporophyte throughout its entire life cycle. Usually the gametophyte is going to be housed on the okay, and then moving on. So like I said, we're mainly focusing on the gymnosperms today, specifically the 3 extant phyla, which are Cycadophyta, Ginkgophyta, and the Coniferophyta. So gymnosperms are a monophyletic group, meaning that it includes its ancestor and all of its descendants. These are the 4 characteristics that I just talked about, the developments in evolution that essentially allowed gymnosperms to become successful and outcompete the basal land plants. So once again, that is the gametophyte being housed on the sporophyte. That is extremely important. As you move on in evolutionary time, you'll find that gametophytes become less and less complex and sporophytes essentially dominate. And then you also have the evolution of seeds and pollen, which are super important. And again, they happened around the same time. And then finally, the evolution of secondary xylem and phloem. So essentially wood and bark. Now, gymnosperms are referred to as naked seed plants because they essentially have naked seeds. So what that pretty much means is that they don't have a fleshy sporophyte tissue surrounding the seeds. So when you think about angiosperms and you think about fruits and flowers, with fruits, the seed is essentially hidden in that really fleshy sporophyte tissue. So the part of the fruit that you actually eat is that fleshy sporophyte tissue. So that's the part that's surrounding the seed. Whereas with gymnosperms, their seeds are pretty much just hidden in the cones and they're completely exposed for the most part. They don't have that fleshy tissue to protect them. So all gymnosperms evolved from heterosporous plant ancestors. So remember that heterosporous means that they produce 2 different spore types, 1 female spore and 1 male spore. So it'll have 2 separate plants, a female plant and a male plant. Specifically megaspores and microspores. So anytime you see the prefix mega, that is always referring to the female, whether it's a megaspore, megagametophyte, megasporangiate cone. Anytime you see mega, it is referring to the female. And anytime you see the prefix micro, that is referring to the male. So microspore, microgametophyte, etc. Anytime you see micro, once again, that is referring to the male structure. They also evolved from plant ancestors that had secondary tissues, most likely, so that's secondary xylem and phloem. Secondary xylem and phloem is super important because it allowed the plants to resist bending and damage and grow a lot taller. As you guys saw in the image back earlier in the presentation, most of the plants were pretty small, like the seedless vascular plants, non-vascular. They really didn't get that tall, but with secondary xylem and phloem, gymnosperms and angiosperms were able to get much taller. Because they were able to get much taller, they were able to get light a lot better, and that means more resources, more productivity as well. And since they were taller, they were also able to disperse their seeds farther away, increasing their chances of reproduction, and they were also longer lived because of secondary tissues as well. Okay, so like I said, with gymnosperms, they are sporophyte dominant. The gametophyte is basically completely reduced and it's housed on the sporophyte, specifically inside the cones. So the gametophyte is really small. You're usually not really gonna see it because most of it is just sporophyte basically. But because the gametophyte is completely reduced, it allowed these plants to survive in drier conditions. So remember that spores are no longer the structure that's actually being dispersed and finishing that life cycle. It's now actually the seeds and the pollen. So because it's seeds now, the spores are essentially obsolete. So once again, they no longer depend on water, and because they're using seeds instead of spores for dispersal, they can disperse and live more inland and at higher elevations as well. Okay, so now we're gonna go over the male cones. So remember that the prefix micro is referring to the male, and then the prefix mega is referring to the female. So this is what the male strobili or microsporangiate cone looks like under a microscope. So I have a better picture of it in the next slide that's a little bit more zoomed in, but I just like the diagram here. So Remember that when I use the term sporophyll, I'm referring to the modified leaf that houses the sporangium. So anytime you see the word phyll, that is referring to the leaf, like how phyllotaxy refers to leaf arrangement, megaphyll, large leaf, microphyll, small leaf, etc. So sporophyll, the microsporophyll here are essentially the scales of the cone. So right here, where that bracket is, this purple area right here, this is all the microsporophyll. The microsporophyll, again, is a modified leaf that houses the microsporangium. So that's this entire circular structure right here. Inside the microsporangium is where meiosis is going to take place, and that's where the male haploid microspores are going to be housed. So just like how with mosses, the sporangium is where the spores were housed, sporangium also known as the capsule. So inside, once again, the microsporangium are the haploid microspores, and those haploid microspores will eventually germinate and produce your microgametophyte or your pollen, or your male gametophyte. Remember, micro means male, so that's where all of that is going to take place. So if you zoom in on the next slide right here, once again, the scale of the cone is the microsporophyll. And then this entire circular structure right here is the microsporangium or the male sporangium. So that's where all the haploid microspores are going to be housed. And then, so these individual little circles, each of those are a microspore, and then those are eventually going to germinate and become your male gametophyte or your pollen grain. But basically this entire sac is your microsporangium. So this is one microsporangium, here's another microsporangium, and then these scales right here are your microsporophylls. So then if you zoom out on that image, again, this like purplish area, that's all microsporophyll. Remember, sporophyll just means a modified leaf that houses the sporangium. And then when you throw micro in front of it, that's just referring to the male. If I said megasporophyll, that would just be referring to the female. Does that make sense? Yeah? Anybody in the chat need me to go over it again, or are we good? Okay, looks like we're all right. Okay, so I'm gonna go ahead and move on. So like I said, in the microsporangium, that's where the haploid microspores are going to be housed. And then eventually those haploid microspores are going to germinate and become your pollen grain, also known as your male gametophyte. Within that pollen grain, there are two different cells. One of them is the generative cell, so that will produce your two sperm or your gametes. And then the other cell in the pollen grain is the tube cell, so that will generate your pollen tube, hence the name tube cell. So again, those are the two cells found within the pollen grain. The pollen grain is really important because this essentially provides protection for the male gametes. And keep in mind, with the pollen grain, it's now going to be dispersed by wind for most of these species. So now, once again, water is no longer needed for dispersal, unlike spores, which required water in order for that life cycle to continue and finish. So once again, pollen grain, you have your tube cell that'll produce your pollen tube. I'll talk about what that does in a second. And then you have your generative cell, which produces your two sperm. Both of these cells are going to be found within your pollen grain. Okay, so the pollen tube, again, from the tube cell. When the pollen grain is being dispersed in the wind, basically what it's trying to do is land on a female ovule or a female cone, essentially. When that pollen grain, or basically the pollen or male gametophyte, lands on a female cone, it'll get a hormonal cue to basically produce that pollen tube. That pollen tube is then going to enter the embryo sac. It's going to burst, and then it's going to release the sperm and it'll fertilize the egg. So if you look at this image right over here, here's your pollen tube and it's carrying those two sperm cells. So once it lands on a female ovule, it'll basically inject itself into the archegonia or the female gametophyte, and then it'll burst, it'll release the sperm, and then it'll fertilize the egg. So that's essentially what the pollen tube does. And again, that comes from the tube cell. The generative cell is what produces the sperm. Does that sound good? Okay, so now we're gonna go on to the female strobili or the female cone. So just to let you guys know, on the lab, it doesn't ask for the anatomy of the female cone, so I'm not testing you on that. This is just to give you a better idea of what it looks like, or to visualize what the seed will look like eventually. So I'm mainly going to be testing you on the male cone anatomy, which is this, and then the anatomy of the seed as well. So you don't have to worry too much about the anatomy of a female cone. But anyways, so female strobili or female cone, same exact thing as the male, where the megasporophylls, so that's the modified leaf, houses the sporangia, in this case, the megasporangia. Anytime you throw the word mega in front, it's just referring to the female. So the megasporophyll houses the megasporangium, just like how the microsporophyll houses the microsporangium. So within the megasporangia, okay, let's backtrack for a second. So let's look at this really quick. This ovuliferous scale right here, that would be your megasporophyll. And then right here at the end, each of these individually is an ovule. Your ovule or your megasporangium is essentially where your megaspores are going to be housed. So if you look at this zoomed in image of an ovule right here, the outermost region, that is the integument. So eventually when your seed is mature, your integument will become your seed coat. That's pretty much like a protection for the seed. And then you have your megasporangium to the inside, your diploid megasporangium. That's going to undergo meiosis and produce your haploid megaspore. That's right there in the center. This is an unfertilized ovule. So inside, here's your megasporophyll once again. That's pretty much the scale of the cone. At the very end of that scale is your ovule. And then here in the center is your megasporangium, and then you have your haploid megaspore in the center. Specifically, it's going to produce 4 haploid megaspores, but only one of them will survive, and that's all enclosed within the ovule. So it'll produce 4, but only one of them will survive. And that one haploid megaspore that survives will eventually become your archegonia or your female gametophyte with your 2 eggs. So just like how the pollen grain produces 2 sperm cells, your archegonia produces 2 eggs as well. But usually only one of the eggs is going to get fertilized, so only one embryo is going to be produced per ovule. So 4 haploid megaspores, only one of them survives to produce your megagametophyte with your 2 eggs. So here's your integument. Once again, this will eventually become the seed coat when your seed is fertilized. Right here in the center is your female gametophyte, your haploid female gametophyte, and then right here is your archegonia with your 2 eggs. This entire structure is the ovule, and that's going to be found at the end of the megasporophyll or your scale, the inside of that. Okay, so now let's talk about pollination. For gymnosperms, most of them are going to be wind pollinated. There are some, for example, like the cycads, that can be animal pollinated, but for the most part, they're going to be pollinated by the wind. So right here on the left, we have a microsporangium. Remember, this entire sac is your microsporangium, and that is where meiosis is going to take place to produce your haploid microspores. Eventually, those haploid microspores are going to germinate and produce your pollen grains or your male gametophytes. So that's what these individual circles are. When that male cone disintegrates, it's going to break open, and then all of this pollen is going to get dispersed in the wind. When it gets dispersed in the wind, hopefully it'll land on a female cone or a megasporangiate cone, specifically the micropyle of the integument. So when I use the term micropyle, I'm essentially referring to the small little opening in the integument. So that's this right here. So when that pollen grain lands on the micropyle of the integument, again, that's just a small little opening in the female cone. When it lands there, that tube cell is going to get a cue to produce the pollen tube. That pollen tube is then going to inject itself into the archegonia or the female gametophyte and fertilize one of these eggs right here. So remember that it produces 2 sperm and the archegonia has 2 eggs, but typically only one of them will get fertilized. So you can see that over here on this image on the right. There's your pollen tube, which is this thin purple cylindrical structure right here. It had inserted itself through the micropyle, which is this opening, and then it's fertilizing only one of the eggs. And then once it fertilizes the egg, then you'll get a mature seed, or basically a new embryo. Okay, but in order for the pollen tubes to essentially insert itself into the archegonia, the cone has to be open. Unfortunately, if the cone is closed, fertilization can't take place. There are some cones that can only open depending on like really high temperatures. Like for example, one of the species that I study, Pinus attenuata, it'll only open their cones in response to fire. Other cones, as soon as it like falls down, there you go, open seeds. But this is essentially what a closed cone looks like, and then an open cone. Okay, so once the seed is mature, there are going to be 3 different generations. You have your diploid seed coat. So remember that the seed coat is from the integument. And then you have your haploid female gametophyte. This is essentially acting as your food supply to the diploid embryo. So again, 3 generations: a diploid seed coat, haploid female gametophyte, which is the food tissue for the diploid embryo. That's that chicken-looking hand in the center. So the seed coat is really important. So remember that this was derived from the integument of the ovule. So the seed coat essentially protects the embryo, and it allows it to go dormant until the environmental conditions are favorable for the embryo to essentially survive. So yeah, 3 generations: diploid seed coat, diploid embryo, haploid female gametophyte. And the haploid female gametophyte is where the embryo is going to take all of its nutrients from. Any questions so far? Thumbs up for good, cool. All right, anybody in the chat? Okay. Okay, so now we're gonna go on to the individual phyla. So the first one that we're gonna be looking at, which is the most basal one, is the cycads, also known as Cycadophyta. Okay, so cycads were dominant during the Mesozoic. They're often referred to as the early gymnosperms because they're the more basal one within this group. They do have monopodial growth, so now we're moving into groups that no longer have dichotomous branching. Some of the individual species do still retain that basal characteristic. For the most part, they're going to be displaying monopodial growth, meaning that they have one main stem that doesn't branch, or it doesn't branch frequently at least. They have really woody stems with a wide pith. So even though cycads have really large and wide stems, the majority of that tissue is actually pith. So it's actually mostly parenchyma rather than secondary xylem. They are evergreen, meaning that they'll keep their leaves on for the entire season. So they have really stiff pinnately compound leaves because of that, and their leaves are actually full of lignin. So remember that lignin is that chemical that helps the cell walls stay really rigid and strong. And the reason why they have a lot of lignin is because they are evergreen, so their leaves need to be able to last a long time, and it also helps prevent freeze-induced cavitation as well. So they do still have flagellated sperm, so their pollen is going to send out a haustorial tube or structure that'll essentially burst when the sperm swim down to find the egg. Most cycads are dioecious, meaning that they have separate male and female plants as well. Okay, so there are 4 different genera that we're going to be looking at within the Cycadophyta. We have Cycas, Zamia, which is my personal favorite, Encephalardos, and Dioun. Now, Cycas and Zamia are the 2 that you should definitely be able to easily tell apart, just because they're pretty distinct from the other 2. Cycas will usually have really thin and narrow leaflets that end in one spine. But another thing that really sets apart Cycas from the other 3 is that they'll always have, I know it's hard to see it in this picture, but they'll always have one kind of like indentation going down the very center of the leaflet, and that indentation is essentially the vein. So one vein or one central vascular bundle going down the center of the leaf. So that's specifically for Cycas. And then we have Zamia over here to the right of that. Now, Zamia is the one that has the rounded edges, so really smooth leaf edges. The leaflets are typically going to be a lot wider as well. And if you were to touch Zamia compared to like Encephalardos or Dioun, Zamia actually has pretty fuzzy leaves as well. So that's one way to tell them apart. The next one is Encephalardos. So this one will usually have more broad or wide leaflets. They're usually going to be about the same size as Zamia, depending on the species, but they will always have spines on the edges of their leaflets as well, whereas Zamia is more smooth and rounded, and Encephalardos will have spines on the edges of their leaves. Now, sometimes with diune, it's kind of hard to tell the difference between diune and encephalitis sometimes, but for the most part, diune is, they're also going to have spines on their leaf edges, but they're usually going to be a lot narrower and thinner in comparison to encephalitis. So that's one way to tell them apart. But personally, I think the easiest to tell apart out of all four of these is cycas and zamia. They're pretty distinct compared to these two. Okay, so now going into the cones for the cycads. So we have the male cones, which are woody and persistent, that are typically not as large as the female cones. Over here on the top right, I have a picture of the male cones. Here's a male cone right here. They're usually going to be a lot longer than the female cones. And then here's another male cone right here in the center of that plant that's starting to form. This is a brand new cone. Now the female cones are bigger and they're ephemeral. So what I mean by ephemeral is that eventually they're going to crumble apart over time. The reason why they crumble apart is so that they can disperse their seeds, which are typically going to be in the center of the plant. So this is what a female cone looks like for a cycad. You can see that it's much bigger compared to the male cone, but the female cones right here are going to break apart eventually so that they can disperse their really colorful seed, which is right here. That's those like large red circles in the cone. And sometimes the female cones can actually also exude nectar to attract pollinators, usually some kind of vertebrate animal. So speaking of vertebrate animals, the cones of cycads are typically going to be a lot brighter and much more colorful to attract these vertebrate animals. Because cycads are typically found in tropical forests where the vegetation is very, very dense, it's actually harder and not as efficient to disperse their seeds through the wind. So because of that, instead, most of the time they're actually pollinated by vertebrate animals. So again, that's why their cones and their seeds are actually a lot more colorful compared to ginkgos and conifers. So these are some examples of really large and colorful female cones. This picture over here on the right is actually taken in the Mesozoic garden on campus. This is from an encephalitis, and it produced these huge female cones, these bright green female cones. And you can see it hasn't opened yet, but if you look where my cursor is, you can see some of the red seeds kind of peeking out a little bit. Okay, so now we're gonna move on to the ginkgos. There is only one species left in this entire phylum. Everything else extinct, gone. Some of you guys are probably familiar with ginkgo biloba because I know that they use this as kind of like a diet supplement. I think it helps for digestion. I'm not 100% sure, but I know that people use this as supplements. Just like Psychedophyta, ginkgos are dioecious, so they have separate male and female plants. But unlike the cycads, ginkgos are deciduous, meaning that they're going to drop their leaves during the winter time, and that's usually to prevent freeze-induced cavitation. So pretty much like air bubbles in the xylem. But they also have flagellated sperm, so their pollen's going to send out a haustorial structure. Usually it's going to be a tube-like structure that will eventually burst to release the sperm and then fertilize the egg. So pretty similar to cycads for the most part, except that they are deciduous, whereas cycads are evergreen. So they do have dimorphic leaves, meaning that they have two different leaf morphologies on the same plant. The leaves of ginkgos that usually have a deeper lobing, those are going to be found on the long shoots, whereas the leaves that have more of like a smooth surface, that's usually going to be found on the short shoots. And I'll show you guys what the long shoot versus the short shoot looks like on the next slide. But again, long shoot leaves will always have that nice, really deep lobing, whereas the short shoot leaves are going to be a lot smoother at the top of the leaf. So you can think of the long shoot leaves as kind of like this main axis right here, this main stem, whereas the short shoots, also known as the dwarf shoots, that's kind of, you can think of them as kind of like the lateral branches. So this is another example of the short shoot right here, and then this would be the long shoot. So again, the short shoot leaves are the ones that have the more smooth or entire leaves. And the short shoots are typically where you're also going to find your female or your male cones. And just like the female cones of cycads, they can also exude a little bit of nectar to attract certain pollinators as well.
I have a question.
Yeah.
You said for the short shoots, they have the smooth and not lobed leaves.
Yeah. So they don't have that really deep lobing in the center of the leaf. So if you look at long shoot leaves, they have that really nice, almost like V-like lobing down in the middle of the leaf, whereas short shoot leaves, they're a lot smoother at the top. So when I use the term entire, that's a term that we use in when we're talking about leaf morphology. It pretty much just means that your leaf is smooth at the top. Does that answer your question? Okay. Okay, so the seeds for ginkgo really resemble a fruit. Oftentimes people will call them a fruit, but fruit did not evolve yet during this time. We don't see true fruit until we get to the angiosperms, which is next week. But the reason why people often think that they are a fruit is because they have a really fleshy seed coat. So usually the seed coats of, for example, conifers, they're usually really hard and rigid, but for ginkgos, they have a really fleshy one. So this like wrinkly structure to the outside, that is the seed coat for ginkgos. And their seed coat actually has 3 layers, 2 hard layers that are to the inside of that really fleshy outer layer. But just like all seeds within the conifers, they have 3 different generations. They have their diploid seed coat, which remember comes from the integument of the ovule. That's essentially to provide protection for the seed. And then they have their haploid female gametophyte, which acts as the nutritive tissue for the diploid embryo. And the diploid embryo is typically going to be in the center. Okay, the last group that we're going to be looking at is the conifers. Now, I'm sure this is the one that you guys are the most familiar with. It is the largest phylum within the gymnosperm. So this includes your pines, your redwoods, and then also some really interesting ones like Podocarpus and Araucaria, which you guys will see in the conifer walk that Dr. Bovich posted. And then the very interesting Nitali, which I'll talk about the last slide. But for the conifers, most of them are actually monoecious, meaning that their male and female cones or their male and female gametophytes are on the same plant. Actually, if you look at most conifer trees, or specifically pines that I know of at least, the male cones are actually usually going to be closer to the base. So they're usually actually going to be lower down to the ground, and the female cones are going to be really high up, and that's to prevent self-fertilization. But they are monoecious, meaning one house. So again, both male and female cones on the same exact plant. However, there are some conifers that are native to the southern hemisphere that are dioecious. Again, that's like Podocarpus and Araucaria, and then the Nitalis are usually dioecious as well. But the conifers that you guys are exposed to here in California, the ones that are native to here at least, most of those are going to be monoecious. So the cones for conifers are the exact opposite of the cycads. This time, the male cones are really, really tiny and ephemeral. So the male cones are the ones that are going to crumble apart and disperse that pollen. And remember that pollen is wind pollinated. And the females are the cones that are going to be much larger. Some can be really, really big. Some are not as big, but the female cones are always going to be much larger compared to the male cones. They're often going to be lignified as well, and they last a lot longer compared to the male cones. The male cones, as soon as you touch it, they're essentially going to disintegrate. And again, that's so that they can disperse the pollen. So these are some other pictures I have of male cones. I believe this male cone is from sugar pine, Pinus lambertiana, and these are from Pinus sylvestris. But again, the male cones are usually going to be very, very small. And where the male cones are, you can usually also see some new growth forming as well, so some new shoots. So that's one example right here. Here are some new needles forming where male cones are. And then here are some more examples of female cones. Right here, all the way to the left, are actually the 2 species that I study. These are pretty dominant in the San Bernardino Mountains. At the top, I have coulter pine, or Pinus coulteri. This cone is crazy. It's also known as the widowmaker because the cones can get really, really large and heavy. And they also have, if you guys can see here, their scales kind of end in like these spines or almost like talons. So they're also called widowmakers because if they land on you, you're dead, I guess, because they can get really big. And they're sharp too. But this is one of the species that I study in the San Bernardino Mountains. And then the second one is right here on the bottom. This one's Pinus attenuata, also known as knobcone pine because they get pretty knobby or sharp. And then we have Pinus lambertiana, sugar pine. These are the female cones. And then a female cone from the coast redwood or Sequoia sempervirens. So bottom line, female cones are always much larger than the male cones for conifers. Okay, so for the conifers, their sperm is no longer flagellated, so they require that pollen tube to reach the eggs. And remember that that pollen tube is going to form from the tube cell of the pollen grain. And the pollen grain, so that they can disperse in the wind and basically travel a lot farther, they have these bladders. I kind of call them like the Mickey Mouse ears that essentially help them float in the wind. So right here where my pointer is and where that black arrow is, these are the bladders, also known as Mickey Mouse ears. And again, that just helps them disperse in the wind a lot easier. So this is over here on the left is a immature male gametophyte. When it lands on a female cone or an ovule, specifically the micropyle of the integuments, remember that's just that small little opening in the ovule. Eventually that tube cell, which is right here, is gonna get a cue to produce that pollen tube. That pollen tube is gonna inject itself into the female gametophyte so that it can fertilize the egg. So even though 2 sperm are going to be produced from the generative cell, which is this cell up at the top right here, usually only one of them will survive, the other one will die. But immature male gametophyte over here on the left, I like to think of that as Mickey Mouse. Over here on the right, we have the mature male gametophyte. I like to think of that as a baby elephant. But they still have the bladders. And then the pollen tube or the elephant trunk is what it's going to actually inject itself into the archegonia. Okay, so now we're going to go into the very weird but very interesting and morphologically diverse, the Nitales. So this is a group within the conifers. Early evolutionary forms were typically lianas or vines, but this group within the conifers is extremely diverse. You have shrubs in this group, you have really large trees, you still have some vines within this order, but very, very diverse. They were actually thought to be the closest sister lineage to the angiosperms or the flowering plants because their male strobili really do look like flowers. And not only that, they also have vessels in their wood for water transport, which is crazy because everything within the gymnosperms only has tracheids until the Nitales came along and they have vessels, which is one reason why they were thought to be the angiosperms or a sister lineage to the angiosperms. But it turns out that they are actually a gymnosperm and they are nestled within the conifers. So in terms of their DNA, they pretty much are a conifer. And then not only that, they also have double fertilization, which is another characteristic that angiosperms have, which results in the production of 2 embryos. So remember that with the pollen grain, it produces 2 sperm cells, and then even in the archegonia, it has 2 eggs as well. So usually with double fertilization, both of those eggs will get fertilized, but one of them still usually dies. But because they have vessels and double fertilization, and they're not closely related to the angiosperms after all, these were a result of convergent evolution, meaning that they have similar characteristics to a group, but because they don't have the same common ancestor, it wasn't a direct lineage. So there are 3 different species that we're going to be looking at within the Nitales. We have Welwitschia. This one only produces 2 leaves during its entire lifetime. And if you see what the plant, I'll show you guys what the plant looked like in a second, but you wouldn't even believe that it was 2 leaves because they get pretty insane. These are native to the Namibian desert in Africa. If you go to the Huntington Garden in Pasadena, I believe they still have a Welwitschia there. If you ever want to go look at it, as soon as this quarantine's over, go to the Huntington because it's pretty crazy. The next one is Ephedra. Ephedra kind of reminds me of Equisitales, which is the horsetails because of the way it grows. But these guys, their anatomy have really obvious vessels. I mean, all Nitales have vessels, but the vessels in Ephedra are pretty obvious compared to these 3. And then we have Needham. This one definitely looks like an angiosperm, especially because it grows like a tree that you would usually see within this group. But they have really broad leaves, and they actually have pinnately compound leaves, unlike some of the other members of the conifer. So this is Welwitschia mirabilis. this only produces 2 leaves. So over here, this is 1 leaf, and then this is another leaf right here. So again, only 2 leaves during its entire lifetime. It looks like there's more than 1 leaf because as the leaves start to grow older and get more mature, it starts to have like tears, but this is actually really 1 leaf and then another leaf. And they also have a basal meristem, which is right here in the center. It kind of looks like that giant hole that actually can also produce wood. And then the brown stuff that you kind of see like shooting up, those would be the cones. But I have a lot more pictures on the slides that you download to answer the questions for Lab 11. There are a lot more pictures for Welwitschia and their cones as well. And then over here we have Ephedra nevadensis. Again, they kind of look like horsetails to me, but yeah, that's Ephedra with their really cool branching. And then this one is Needham. So like I said, it really does look like an angiosperm. Even their cones look like actual fruits, but these are essentially their cones. Again, they're not true fruit because they are within the conifers, so no fruit just yet until we get into the angiosperms. But yeah, that's everything for this lecture. I did record this, so I'll post this on YouTube and then send you guys a link because unfortunately I have met my quota on Blackboard. So let me stop this share really quick. So just so you guys know, I had to delete all of the old PowerPoints on Blackboard to make room for the ones that you guys actually need for the final practical. So you won't see the PowerPoints for labs 1 through 9 on there because I'm only testing you on labs 10 through 12 for the practical. If you wanted any of the PowerPoints from labs 1 through 9, like you were never able to download them or something, just send me an email and I can go ahead and send that to you, but I had to delete it to make room for the PowerPoints that you actually need to study for. So yeah, so that's all I have for you guys. The PowerPoint for Lab 11 is already up. Don't forget to take your pre-lab quiz, and then you guys will turn in the assignment for this lab next Wednesday at the same time. But yeah, that's pretty much all I have for you guys. You guys can leave if you want to. You can always log off and then log back on if you guys have any questions. But yeah, I will be here. So